New hope for the incurable diseases in the aging population. Curacle. Curacle, a dreaming next-generation bio company, has developed the innovative therapeutic agent based on blocking vascular endothelial dysfunction to lead the global market. Curacle aims to develop the innovative new drugs that benefit the society and contribute to the enjoyment of the healthy and happy life for mankind. Blood vessels in the spotlight as the key to disease-free long life. Particularly, capillaries are emerging as the hotspot for the next generation drug development. Capillaries, occupying 95% of the 10-kilometer-long blood vessels of the human body, are the passageways that supply oxygen and nutrients to all cells and tissues and dispose of the waste products. Recently, many scientific studies have revealed that various intractable metabolic and inflammatory diseases start from the destruction and dysfunction of capillaries. Hyperlipidemia and antithrombotic agents centered on the coronary arteries, which are viewed as the first generation of vascular therapy, have been on the market since the late 1980s. After this, a treatment targeting the growth factors of capillary blood vessels, which corresponds to the second generation, began to attract attention. Since the late 2010s, a new paradigm of the therapeutic development has been attempted, centering on Curacle. This third-generation new drug is expected to change the paradigm of a new drug development for the vascular diseases, because it is a new concept that directly targets capillaries, the cause of the intractable diseases that could not be treated with a single target, and treats the capillaries with multiple actions. Curacle Sovatis is a unique platform specializing in the development of third-generation vascular-related innovative new drugs that block the damage to the vascular endothelial cells through the systematic phenotype-based screening. Various kinds of active substances that cause diabetes, aging, obesity, infection, high blood pressure, allergies, and free radicals, etc. Destroy the vascular endothelial barrier. Introduce inflammatory cells into the tissue. And cause fetal disease through the death of endothelial cells. CU06, developed by Curacle, shows excellent results in blocking the vascular leakage and tissue inflammation through the restoration of the structure and function of the vascular endothelium by acting specifically on the vascular endothelial cell membrane. This drug will be expanded as a checkpoint treatment for the various intractable diseases and is expected to dramatically improve the treatment efficacy and convenience for the patients. Along with this, Curacle's pipeline of the innovative new drugs under research and development will open a new path to cure the diseases traditionally considered incurable. And Curacle's dream and challenge for human health to reach the age of 150 will continue.
Curacle is a Korean R&D-focused biopharmaceutical company, and we aim to develop the next generation treatments for vascular-related intractable diseases. Our main asset is an endothelial dysfunction blocker derived from the proprietary drug discovery platform Sovatis. Its efficacy has been proven in 8 animal model studies for 8 different diseases, such as macular edema, acute lung injury, myocardial infarction. We possess a strong R&D capability and received a Fast Track designation for an innovative tech company. I will start by introducing Curacle, then move on to our unique platform Sovatis and discuss each of our pipelines in detail. Curacle was listed in the Korea stock market in July of last year, just 5 years after being founded. In this process, we received a AA grade in technology evaluation from the government. And last October, we completed a global licensing deal with a total deal value of $2 billion with DEA, a leading independent ophthalmology company in Europe. The deal was for our molecule CU06 with the indications of diabetic macular edema and wet AMD. Curacle's founder, Professor Kwon, is a dean at Yonsei University, a top private university in Korea. He is considered a top expert in the field of vascular disease internationally and has more than 3 decades of experience. This is a brief overview of Curacle. We are an R&D focused company with 67% of our workforce being R&D specialists. We have the best experts in the field for scientific advisors covering diverse therapeutic areas. The members in the yellow boxes below are the SAB members for the co-developing of the project with Thea for phase 2 clinical study in the US for ophthalmology indications. So here is the snapshot of our pipeline and its stage of development. 3 pipelines, CEO6, CEO1, and CEO3 are under clinical trials. CEO6 is a substance derived from our platform Solvetis, and it is a first-in-class drug for DME and wet AMD. It's been licensed out globally, except for the Asian regions, and is still open for licensing out in the Asian market, including Greater China and Japan. CU01 is dimethyl fumarate with the new indication of diabetic nephropathy. We completed phase 2A clinical trial in Korea, and clinical results show statistically significant improvement of eGFR, and no SAE was reported. We have submitted IND for phase 3 end of last year. CU03 is an oral drug for combination therapy for wet AMD. We also have ongoing projects, CU101 through 105, as indication expansions of CU06 for myocardial infarction, acute lung injury, stroke, ulcerative colitis, and oncology combination. CEO4 and CEO5 are oncology pipelines. I'll discuss our platform Solvetis used for developing endothelial dysfunction blockers. The capillary vessels, which compose 95% of the blood supply of our body, provide oxygen and nutrients to all cells and tissues. But as capillaries degenerate with age, it leads to vascular-related diseases and metabolic diseases. Vascular endothelial dysfunction is mainly caused by vascular leakage, in which the endothelial cell junction is broken and increases the expression of inflammatory cell adhesion proteins. Aging, diabetes, and infection stimulate endothelial cells, and changes endothelial cell functions leads to edema and tissue inflammation. These 8 diseases listed here are related to endothelial cell dysfunctions. We develop first-in-class treatments for various intractable vascular diseases using primary human endothelial cells and disease phenotype screening. Using Solvetis platform, we can screen for multifunctional candidates that are low in toxicity and high in safety. Through the animal model studies, we found that CEO6 has efficacy in 8 diseases. CU06 is a first-in-class endothelial dysfunction blocker for diabetic macular edema, and we are currently looking for partners in the Asian region. It targets multiple factors, and it was developed through the platform Solvetis. It is the first oral drug that can solve the unmet needs of anti-VEGF treatments by providing the convenience of administration to ease the discomfort of injection to the eyes, multi-factor working mechanism of action to overcome non-respondents to anti-VEGF treatments, and applicability for a broad range of patients. In animal tests for retinal vascular disease using beagles, CEO6 reduced macular edema by 48%, comparable with the efficacy of Eylea injection, which reduced edema by 35%, and effectively inhibited abnormal angiogenesis. It is currently under phase 1 in the US, and we plan to enter phase 2 this year. Our ongoing projects are for indications of myocardial infarction, acute lung injury, and stroke, as our deal with Thea is limited to ophthalmology indications. These projects are available for out-licensing. The current treatment for MI is to minimize the infarct area by recanalizing the coronary artery. However, edema, bleeding, and inflammation caused by oxidative stress during the restoration of blood flow account for 30 to 40% of the myocardial damage. Acute lung injury is caused by many factors such as inhalation of toxic substances, sepsis, or COVID. In this process, damages in vascular endothelium and alveolar epithelium leads to lung edema and inflammation. CU102, an endothelial dysfunction blocker, can be a treatment option for acute lung injury. In stroke patients, blood-brain barrier breakdown and dysfunction have a major role in neuronal loss and brain damage. CU103 maintains blood-brain barrier integrity and provides neuroprotection. For the three indications, efficacy has been proven in preclinical studies. CU01 is a first-in-class drug for diabetic nephropathy that improves renal functions. Currently, there is no fundamental treatment for diabetic nephropathy, and treatments for hypertension and diabetes are being used with some limitations. However, this doesn't treat kidney fibrosis. CU01 is a new indication of dimethyl fumarate for diabetic nephropathy. Its MOA is to inhibit renal fibrosis by activating NRF2 and inhibiting TGF-beta and SMED3 signaling pathway. DMF stimulates antioxidant and anti-inflammatory actions while inhibiting fibrosis. We completed phase 2A study in Korea in March of last year, and CU01 showed very successful results in clinical trials conducted on patients with diabetic nephropathy. The dose was 120 milligrams, half the maintenance dosage of Tecfidera. The results show that the eGFR, a representative indicator of renal function, increased in the treatment group, and the findings were statistically significant. There were no SAEs or deaths related to the test drug, and no AEs were reported other than known AEs of Tecfidera. We expect to begin clinical trial phase 3 early this year. You can see the details of our CU01 patents here and their status. The scope of our patent is very comprehensive, including patents for indication, formulation, and dose. CU03 is a first-in-class drug for wet AMD. It is an oral botanical medicine for wet AMD, and it's multi-acting through anti-inflammation, anti-angiogenesis, and antioxidative properties. The efficacy for these three MOA has been proven in preclinical studies. We were able to identify more than 90% of the indicator components, and their chemical structures were characterized. In vivo efficacy of 12 marker compounds have been validated. Our phase 2A clinical study showed no tolerability issues, and there were no SADR or SAEs related to the drug. CMT was reduced dose-dependently in the group that received the rescue drug Eylea. It is being developed as a combination therapy with anti-VEGF to increase the overall efficacy and reduce the frequency of anti-VEGF injection. And it is also applicable to early-stage patients. We plan to carry out phase 2B study in the fourth quarter of this year. I will briefly discuss our oncology pipeline in the discovery stages. CU04 and CU05 are currently in the validation stage of the preclinical candidates. CU05 is a first-in-class AKT pathway inhibitor. It shows efficacy against a broad range of solid tumors in the lung, colorectal, breast, and pancreas. 3 final candidate compounds have been confirmed. We are evaluating the final candidates with in vivo, PK, and toxicology results, and we expect to enter the preclinical study this year. This is the overall status of patents of our assets. We have secured a total of 72 intellectual property rights at home and abroad by registering for 17 patents and applying for 43. Thank you for watching.